Hello, my name is Alan Hudson from the Immuno-Oncology Translational Network Data Management Resource Center. I also serve as Chair of Biostatistics and Bioinformatics at Roswell Park Comprehensive Cancer Center. Before we start the seminar, I just have a few bookkeeping items to go over. Questions will be responded to at the end of the presentation. Please enter your questions in the Zoom Q&A box, not the chat box, the Q&A box. The audience can prioritize the order of the questions by clicking on the thumbs up button. Closed captioning is available. The closed captioning for today's webinar can be accessed by clicking on the live transcript option in the Zoom menu at the bottom of your screen. Instructions will also be provided in the chat box. If you're having technical issues, you can make a comment in the Zoom chat box. With that, I would like to turn the proceedings over to our moderator, Dr. Eduardo Villar-Sanchez, who is Associate Professor and Deputy Chair of the Department of Cancer Prevention at MD Anderson Cancer Center. Dr. Villar-Sanchez's research efforts are focused on the development of novel cancer interception strategies, including vaccines and chemopreventive agents, and early detection for patients at high risk for colorectal cancer development. The Villar-Sanchez Laboratory has characterized the genomic and transcriptomic landscape of precancers using next-gen sequencing approaches and has identified novel targets for interception using system biology tools. In parallel, his research team has applied systematically the same workflow to genetically engineered mouse models that mimic the natural history of hereditary colorectal cancer syndromes to perform cross-species comparisons and validate these novel preventive agents and biomarkers in vivo. Dr. Villar-Sanchez has designed, implemented, and participated in several investigator-initiated clinical trials for early drug development and cancer prevention in hereditary colorectal cancer populations that have been funded by the Division of Cancer Prevention at NCI and as well as industry. Eduardo?
Well, thank you so much. Welcome everybody to the Moonshot Seminar Series. And today I have the distinct pleasure to introduce our speaker, Dr. Sean DeMary. Dr. DeMary is an associate professor in the Center for Cancer Immunology and Cutaneous Biology Research Center at Massachusetts General Hospital and Harvard University Harvard Medical School. Dr. DeMary received his undergraduate degree in biology from Washington State University, and then he completed a medical degree and a PhD in cell biology in the laboratory of Dr. Raphael Copen and a dermatology residency training at Washington University in St. Louis. Dr. DeMary then conducted postdoctoral research in immunology in the lab of Dr. Wayne Yokoyama at Washington University. Dr. DeMary is a physician scientist performing cutting-edge basic research and translational research in the field of cancer immunoprevention, as you are going to see in his presentation today. Related to skin and breast and other epithelial cancers. Today's presentation, as you can see in the title slide, will focus in breast. His research has the overarching goal on determining the role of the immune system in maintaining normal tissue homeostasis and preventing the earliest stages of cancer development. Dr. DeMary has a very distinguished career, is a recipient of several awards that include the American Academy of Dermatology Young Investigator Award, the American Society for Clinical Investigation Young Physician Scientist Award. Currently this society, and he's a current member of the Society of the ASCI. He also was awarded the NIH Director's Early Independence Award, the Burroughs Wellcome Fund Career Award for Medical Scientists, the Breast Cancer Alliance Young Investigator Award, a Kimmel Scholar Award, and a Leo Foundation Award. So as you can see, Dr. DeMary is highly distinguished. He has many publications in high impact factor journals under his belt, and on the personal note, he's a great person, a great collaborator, and a great friend. So welcome, Sean, and looking forward to your talk.
Thank you so much for the very kind introduction. I'm going to basically try to jump into the talk, and hopefully this would be if you will, a unique and interesting area of immunology as we discovered the interaction of the immune cells with the early malignant cells within the breast in this case. So without further ado, our lab is interested, as mentioned, in the question of immune regulation of early carcinogenesis, mostly in the context of epithelial cancers, where we can detect, in many cases, early malignant clones and early lesions, what we call pre-malignant lesions in the epithelium. And our lab has been extensively looking at this question of epithelial regulation of cancer development. And as one of the factors, apart from, if you will, antigens that could obviously be recognized by T cells and for control and regulation of this development, we do have what we call immunomodulatory agents, and namely epithelium-derived cytokines and alarmins that we believe may play an important role in this process of cancer recognition by the immune system. So among these epithelial-derived alarmins, my laboratory has been very interested in type 2 alarmins. So these are thymic, stromal, lymphopoietin, IL-33, and IL-25. And as you probably know, these cytokines are mostly studied in the context of allergic inflammation in the skin, in the lung, and other barrier organs. However, if you step back and think about these cytokines, their function is really described, or their induction, if you will, is described as a consequence of defective barrier formation in many cases in many of these barrier organs. And what we believe, at least what we're seeing in the studies we have conducted, is that when a similar aberrant differentiation happens during malignant transformation, you may be, the tissue may be experiencing some of the same signals from these cytokines. And we were really interested to understand what those consequences of those inductions are in various organs. We have done a lot of work on TSLP in the skin, as mentioned, and also in the breast, but in the skin, we have actually taken this from a very basic discovery to now to a topical agent that induces this cytokine, and it's on NCCN guidelines for treatment of pre-malignant lesions in the skin as perhaps the first T-cell-directed immunotherapy for cancer prevention in the skin. The talk today really relates to the role of this cytokine as it relates to breast cancer immunoprevention, and the original reason for that was the findings from us and other groups that the cytokine is present within the breast gland, both in mice and humans, and also for us to understand how this cytokine really regulates immunity in a non-barrier organ, which is breast. And this work has been led by Dr. Margarita Boeri in my laboratory, talented postdoc who spearheaded most of the data that we'll be presenting in this talk. So as I mentioned briefly, TSLP, or thymic stromal lymphopoietin, is a cytokine mostly studied in context of allergic inflammation. It can be expressed by epithelial cells and other cell types as well, but classically in context of allergic inflammation, we believe it has been shown that allergens exposure and, again, aberrant differentiation of the epithelial leads to the expression of this cytokine. This cytokine primarily induces a type 2 immune response at the barrier sites, by promoting Th2 differentiation and also ILC2 differentiation at these sites against these, if you will, allergen exposure, and leading to asthma, atopic dermatitis, food allergy, and other allergic disorders. When you look at the cytokine, TSLP cytokine expression in the cancer versus normal breast glands in humans from TCGA GTEx data sets, you can appreciate immediately that there's a loss of this cytokine expression within the tumors compared to normal glands. And we validated this also looking at the protein from the whole lysates of the breast tumors and their adjacent normal glands. And we can see that there is a reduction of global TSLP levels at the protein level in these tumors. And interestingly, if you then look at the various stages of tumor development in the breast, from perilesional normal to carcinoma in situ and then invasive carcinoma, there's a gradual loss of the expression of TSLP that is, again, mainly coming actually from epithelial cells in this organ as well. When we look at although there's levels of TSLP in the cancer is markedly reduced, but you can still distinguish the population of patients who have higher expression of TSLP versus the low expressors. And you can see the survival benefit for folks that have higher levels of TSLP expressed in their breast tumors. So this set of data has really prompted us to look at this question of TSLP induction in context of breast cancer development. And what does that do if we restore TSLP during the carcinogenesis process and understand what happens to the tumor development in the host? And to do so, we have been taking advantage of genetic approaches to do this. So this here we are showing you the mice, again, the wild type animals with their breast gland, and it's pretty much a very cold tissue environment, not many T cells or other immune cells around these glands in the mice, and the branching is perfectly fine. If you overexpress TSLP to systemically, if you will, in these animals, the picture is very much the same. There's minimal amount of immune infiltrate, and branching of the glands are perfectly normal in a nulliparous mouse. If you will look at the genetic model of breast carcinogenesis, in this case, PYMT or MMTV PYMT, which spontaneously develops tumors, these mice develop early tumors early on. I believe this is 10 weeks of age, and you can appreciate this in situ lesions in their breast. Again, very much cold, not immune infiltrated. However, if you now look at mice that both express the oncogene in their breast, but also overexpress TSLP systemically, you have a very distinct alteration of these pre-malignant lesions, as you can see here, with infiltration of T cells. And interestingly, at the endpoint, so this pictures the carmine stains are at the endpoint of tumors when the PYMT mice have reached their terminal size. We do see initiation of tumor development in the TSLP PYMT, but tumors, even at this level of magnification, look arrested in their development and not forming aggressive large tumors. When you quantify these immune cell types at the end of the study, in terms of when the control groups reach their terminal tumor size, the test group, which is protected, we believe, from tumor, have many immune cells in them, mostly CD4 positive T cells, but to some degree CD8 positive T cells also accumulating in these tumors. As I mentioned, the tumor onset is delayed in TSLP PYMT mice compared to PYMT animals. They develop significantly fewer the volume of tumors, if you will, at the endpoint, and then their survival is markedly extended compared to the PYMT control mice. But above all of this, and again, that's really the survival and the onset relates back to the tumor growth rate and the tumor size over time. What we discovered, which was quite surprising in the field, was not just that you're controlling the growth of these tumors, but in fact, you're altering the phenotype of these tumors from the TSLP induction axis. In other words, the tumors that normally develop in mice that have the oncogene PYMT end up being what we call high-grade tumors with solid sheet of tumor cells covering the tumor area. When now you have TSLP in the environment, beyond the tumors being much smaller, they actually histologically look very different, and they look differentiated. In other words, in many cases, we observe these cystic structures in place of the high-grade tumors in mice. And if you quantify this across mice that we study, the spontaneous tumor model mice in PYMT backgrounds, you can appreciate this distinct difference in tumor grades of these animals compared to the control mice. And perhaps this is a nice way of looking at it. So these T cells that we see infiltrating into these tumors of TSLP PYMT, they're encompassing these glandular structures. In other words, they're arresting these tumors into what looks like fibrocystic-like structures and don't allowing them to progress to high-grade tumor development in the animals. So we wondered what causes this, if you will, block or suppression of tumor development. Classically, obviously, we think of cytotoxicity to be the mediator of anti-tumor immunity, but we don't really see any of that effect in these mice, in these tumors. Various ways of looking at this question of apoptosis and cell death did not really result in any distinction between the test and control mice. However, when you look at the proliferation of the tumor cells, again, this is an early time point of tumor development. We can clearly distinguish the test group of mice that are exposed to TSLP compared to the control animals, which are basically having very proliferative tumor foci in their breast. And again, you can also appreciate in the background, it's not just they're less proliferating, they're actually looking at the gland here, almost like all these cystic structures versus a very solid grouped cells in the context of PYMT itself at this age of tumor development. So we took these tumors and performed RNA sequencing on them to understand what exactly is going on with the tumor cellular phenotype of these tumors. And this is quite obvious from this analysis that the tumors that are experiencing TSLP signal are basically differentiating. So these are undergoing terminal differentiation with breast gland protein expression in them, like beta-casein and alpha-lactoalbumin and others. However, on the other side, if the control tumors are expressing genes associated with tumor progression in the breast. So this was quite a novel, interesting phenotype that we observed. Again, histologically, just from an H&E perspective, was quite striking how these tumors change their, if you will, their morphology as they're exposed to TSLP. This is just demonstrating, again, another way of looking at this question of differentiation of the tumor cells, that TSLP-PYMT tumors express genes and proteins associated with differentiation of breast gland versus the PYMT tumors expressing these EMT-related gene expression profiles in their tumors. So to understand this more at the immunological level, what's going on and what is really driving this phenotype downstream of TSLP induction, we resort to immunological assays, which primarily rely on tumor transfers and T cell transfers to understand this phenomenon. So we took recipient mice. These are syngeneic mice that are overexpressing TSLP but don't have any TSLP receptor in them, and then transferred into them congenetically marked wild type or TSLP receptor knockout CD4 T cells. Following to that T cell transfer, we then transfer tumor cells into these mice, which also don't have any receptors on them. So these are PYMT transgenic mice that lack TSLP receptors. So in this whole combination of mice and tumor and T cells, the only cell that could receive TSLP signal would be the test wild type CD4 positive T cells. The rest of the environment is deficient in the receptor and blind to TSLP signal. So this would allow us to really hone in on CD4 T cell immunity and understand what the role of that immunity is in this effect that we're observing in the breast cancer. So if we do this experiment using, again, the PYMT tumors that lack TSLP receptor and comparing, again, the wild type T cells transferred versus TSLP or knockout T cell transfers, you see that there's a protection just by having a wild type T cell, CD4 T cells in the environment. You can deliver protection from TSLP against the tumor development in the mice. This protection actually can be amplified if you provide a very prototypical antigen, OVA, to be expressed with the PYMT in the tumor cells. You can really amplify this effect to a great degree, suggesting that there is the levels of antigen specificity in this process. And I'm not showing that data here, but I would also like to mention that we've moved from this to look at the question of antigen-specific T cells, and we can demonstrate that you really need those antigen-specific T cells to deliver this protection. And for example, OT2 or CD4 positive T cells protect against PYMT OVA tumors upon induction of TSLP, but these same OT2s, for example, cannot protect against PYMT and vice versa, so you can demonstrate the antigen specificity of the phenotype we observe. Next, we moved on to looked at the survival again, especially in the PYMT OVA TSLP receptor knockout tumors that are exposed now to wild type CD4 T cells, and we can demonstrate that there is extended survival in these mice associated, again, interestingly, to lower grade of tumors that do exist in this in the breast gland of these mice after tumor transfers. So this picture here demonstrates, so these are the donor tumors that came from the PYMT OVA TSLP or knockout mouse and went into the, again, mice that are either have wild type CD4s or TSLP or knockout CD4s. You can appreciate that within the wild type, the mouse that has wild type CD4 T cells, these tumors that are developing they're having a very distinct morphological and grade, histological grade, if you will, compared to the control animals. If we look at these tumors now that are developing in a recipient mice, the same observation is made as we saw in the spontaneous tumors associated with the massive induction of CD4 positive T cells, some CD8s, but mostly CD4 positive T cells in the tumor microenvironment, and also development of some tissue resident memory CD4 positive T cells in this environment as well. So we went on to profile these CD4 positive T cells, and as you can expect from the function of TSLP on direct function of TSLP on TSLP receptor on CD4 T cells, it's really known to drive a Th2 phenotype, a type 2 T helper 2 type phenotype from the CD4 polarization. So that's what we're seeing here as well. The majority of these congenetically marked wild-type CD4 T cells in the tumor microenvironment are becoming Th2 cells. And these are really the cells that are dividing within the tumor microenvironment. So these are the GATA3 positive, Ki67 positive cells in the tumor microenvironment. So we next asked a very specific question that relates to this, again, another novel aspect of this discovery, which is we are dealing with not only CD4 T cells that are differentiating perhaps the tumor cells, but also these CD4 T cells are of Th2 phenotype. So is Th2 polarization required for TSLP-induced CD4 positive T cell immunity against breast carcinogenesis? And to address this question, we had to go back to genetic models of cancer development or the spontaneous tumorigenesis and remove IL-4 receptor from the environment in mice and understand if removal of IL-4 receptor, which required for Th2 polarization, would abrogate any phenotype in terms of protective effect of TSLP. So now in this figure, you're looking at the control groups. So the PYMT, which is in black, and you can see the time to tumor onset and survival of these mice over time. TSLP-PYMT, which is again another positive control group that I showed previously, and those mice are protected against breast cancer development, both in terms of the time to tumor development and the survival of the mice. And also the PYMT IL-4 receptor knockout, which is surprising to us, had even faster tumor onset compared to the PYMT mice, as you can see here. So in addition to this, now if you look at the test mice that we were really wanting to study, which are the TSLP-PYMT mice that lack IL-4 receptor in them, you can see that these mice behave very much like PYMT IL-4 receptor knockout. They basically have lost any effect of TSLP in them or any protective effect of TSLP in them, strongly demonstrating that really this phenotype that we're observing in mice that is caused by TSLP induction really requires Th2 polarization for its efficacy in the animals. So we next wanted to understand more of a, if you will, a therapeutic angle to this, as was mentioned in context of cancer immunoprevention. Can a transient TSLP induction suppress breast tumorigenesis? And to look at this question, we took advantage of a very well-established system to induce TSLP systemically in mice in a short interval, if you will. And that's the use of topical calcipotriol or MC903. This is a chemical, a vitamin D, a low calcimic vitamin D analog that obviously binds to the VDR vitamin D receptor, and vitamin D receptor has binding sites on the promoter region of TSLP. This system is very much often used for induction of TSLP from the skin in the context of studies relating to allergic inflammation, atopic dermatitis. But when this application is performed, you can appreciate induction in serum TSLP levels that get systemic in the mice. And this way you can look at this question of systemic induction, but in a very short window of time to understand how that impacts tumor development in the breast. So in this context, to do this type of analysis and this type of experiments, we are now taking advantage of just TSLP receptor knockout mice without any genetic induction of TSLP. Otherwise, the cells are very much the same. So T cells would be either wild-type T cells or TSLP receptor knockout CD4 positive T cells injected or transferred into these mice. And then the mice receive, in this case, PYMT OVA TSLP or knockout T cells, tumor cells. And then after the tumor transfer, the mice are treated with topical calcipotriol to induce TSLP expression. So in the first set of analysis, we looked at the induction of TSLP at the early time point after the tumor transfer. So as soon as the tumors were transferred into mice, 2 days after that tumor transfer, the mice received the calcipotriol treatment for 30 days, and then the treatment was stopped, and we followed the mice over time to understand what happens to their tumor kinetics over time. And this is the outcome of this study. It's a very obvious inhibition of tumor growth, which persists, at least for the follow-up that we have done over a month after you stop the treatment with calcipotriol in these mice. that are having, again, wild type CD4 T cells and everything else being equal in the animals. This extended lifespan, again, associates with extended survival. And these mice, again, go on not to have any tumor growth after the stop of the treatment. And their tumors, unlike what, again, we see in the context of cytotoxic immunity, in this context, the tumors or the node, if you will, or the lesion that remains behind looks very much like a fibrocystic structure with a lot of fibrosis, and these, if you will, almost differentiated breast gland structures within the site that the tumor was delivered in the mammary gland. And that's what we see, again, if you extend this even out for 2-3 months after the stop of the treatment in the subgroup of these mice. So it's a persistent differentiated state of the tumor, which is quite interesting and distinct from cytotoxic immunity against epithelial cells. Next, we examine whether induction of TSLP after the tumor has been established in the mice could still deliver this type of differentiation phenotype and tumor arrest phenotype that we observed. And to do this experiment, we hit the same platform of tumor transfer and T cell transfers. However, in this case, we waited for the tumors to reach a palpable size of 5 millimeters in diameter and then treated them with calcipotriol for 30 days. And as you can see here, this analysis, this study very much phenocopies the early treatment with calcipotriol, and the mice that do respond to this treatment, they have very much protected status and no tumor growth after we stop treating them with the TSLP induction. And this extends again for months after that, and pretty much the same phenotype of tumor differentiation. We still can appreciate, again, a node or if you will, a very small lesion at the site that these tumor cells were delivered into the mice, but these tumor cells very much look like in an arrested phase of differentiation and not having any potential to progress to advanced cancers. I also should mention that in these tumors, in these foci, if you will, at the end of the study, even though this is 1 or 2 months after the treatment, you can appreciate the presence of T cells in this microenvironment in these test tumors compared to the controls. So then we wanted to understand obviously more deeply what is really going on. So we so far can demonstrate that TSLP induction, by binding even directly to CD4 positive T cells, delivers this pro-differentiation effect against tumor cells, both blocking tumor development by induction of terminal differentiation, but also by reverting already established high-grade tumors to revert back to fibrocystic-like structures in the animals. But can this be done directly by CD4 positive T cells? And that's really obviously a key question in this process, especially when we are talking about here Th2 cells, and how can they really be effector cells in this context? To address this, we first looked at the question, the fundamental question of where is this antigenic presentation from the tumors come to be detected by T cells? Do breast cancer cells express HLA class 2 or MHC class 2? And they don't. We don't really see much of expression on epithelial cells in the context of mouse models of tumors, in this case PYMT. However, when you have TSLP induction in the microenvironment, you have very, very high number of antigen-presenting cells. And we believe most of these are macrophages, but also dendritic cells, these CDC1s that are presenting antigen to T cells in this context. So this is not a direct contact between the T cells, CD4 T cells, and tumor cells. However, there is plenty of space for T cell activation with this microenvironment to these tumors that are experiencing TSLP induction. So we went on to specifically try to address the question of, can CD4 T cells that are not directly contacting tumor cells have direct effect on tumor cells and differentiate them and cause their arrest in their growth and proliferation? To do this analysis, we took test mice, these are TSLP PYMT tumors, versus control tumors, which are PYMT TSLP knockout, and isolated CD4 T cells from these tumors and their lymph nodes and expanded them in culture with CD328 and TSLP cytokine. And then isolated their supernatant and used that supernatant on normal mammary gland epithelial cells from mice called HC11 in the mammosphere growth platform to understand if this supernatant coming from CD4 T cells that can experience TSLP versus those T cells that cannot experience TSLP because they're TSLP receptor deficient would have differential effect on these mammosphere growth in culture. And the answer is that the effect is quite actually strong. So if the test T cells, so these are basically wild-type CD4 T cells from a tumor that was experiencing a lot of TSLP, and again, in the culture got exposed to TSLP as well, the supernatant from these T cells is very much inhibiting the growth of the mammospheres, actually also differentiates them into more spherical looking mammospheres compared to the control supernatant, but also control media groups. This is a quantification of the size of these mammospheres. But again, beyond the size, we do see a very nice effect on differentiation of these mammospheres into more spherical shaped and hollow center, if you will, mammospheres compared to the almost like a mass of cells with no architecture in the context of control T cell supernatants and also controlled media exposures. So this allowed us to at least understand that as TSLP activates CD4 T cells, these T cells directly could block tumor development, and they can actually directly affect the epithelial cells in the breast. So what cytokines or what immune factors could be coming from these TSLP-activated CD4 T cells that drive this phenotype? To address this question, we performed various analyses over the years, and I'm just highlighting a couple of these. One is obviously RNA sequencing to look for what factors these activated CD4 T cells are expressing. As you can imagine, there are CD4 T cell Th2 cells, so they have hallmarks of that expression with IL-5 and IL-4 and IL-13 overexpressed in them. But we also appreciated a group of cytokines here, the CSF2 or GM-CSF, IL-5 and IL-3, that are being expressed by these Th2 cells at a very high level in these mice. We validated these actually using secretome proteomics, trying to understand what secreted proteins could be released by TSLP-activated CD4 positive T cells, and we arrived at the same group of cytokines at top of the list of secretome from the T cells, and then validated that using ELISA assays I'm showing you here, looking at IL-13 as the hallmark of Th2 differentiation, but also IL-3, IL-5, and GM-CSF. So that basically suggested to us that perhaps there might be a role for these cytokines in this effect that we are observing in mice in control of breast tumor development. And we asked that question specifically, what is the role of IL-3, 5, and GM-CSF in suppressing breast tumorigenesis? To test this question, we went back to our tumor transplant model that I previously shown, which is mice. These are TSLP receptor knockout mice that overexpress TSLP. And then you give them tumors which are PYMT ova, TSLP-R knockout tumors. And at the same time, you give them T cells, the CD4 T cells are either wild-type CD4 positive or TSLP-R knockout. And this is the difference we saw when we did that experiment with wild-type CD4 T cells. In this context, we are now doing the same experiment. Again, the recipients overexpress TSLP, don't have receptor. The tumor does not have receptor. And then the only thing that has the receptor for TSLP is, in this case, test T cells which lack IL-3 expression. And we are comparing them to TSLP-R knockout CD4 T cells as controls. At the same time, we are also treating the mice with IL-5 and GM-CSF antibodies. So we are using IL-3 knockout CD4s and block IL-5 and GM-CSF, and we are comparing them to TSLP-R knockout. And in this context, we lose any protective effect of TSLP, as you can see in this tumor growth spider plot. So that allowed us to make the understanding that TSLP activation of Th2 cells lead to these cytokines, and these cytokines playing a critical role. They're required for the tumor protective effect that we're seeing in the mice. However, the big question that still was remaining is that, is this really are these direct cytokines directly mediating this effect on tumor cells, or they're perhaps activating other immune cell types, eosinophils, other cell types that could respond to these cytokines and classically are more known to be responding to these cytokines. And they might be coming here into the tumor and function as effector cells, mediating the effect of T cells onto the tumor cells. So to address this question, we went back to the mice and we looked at, or mouse actually in humans, and looked to understand if these receptors are expressed, receptors for IL-4, IL-5, IL-3, and GM-CSF are expressed on epithelial cells in the breast. And the answer is yes. So the common receptor between all of them, which is common beta chain, is certainly expressed both in mice tumors, but also you can appreciate the expression in the RNA data from humans, and you can stain for that. In this case, we're staining for IL-3 receptor in the breast glands and breast tumors of the patients, and you can appreciate the expression of this at the protein level on the epithelial cells in the tumors and in the glands. So there is a very plausible scenario here that these cytokines are directly mediating the effect from CD4 positive T cells onto the tumor cells themselves. And that would also correspond to the in vitro mammosphere data that we had that should demonstrate that CD4 T cells can directly function as effector cells and inhibit the tumor growth and mammosphere the growth in that case. So to finally address this question, we had to generate mice that develop tumors, PYM2 ova tumors, that lack the receptors, common beta chain and IL-3 receptor beta-2, which is the arm of the IL-3 receptor that is distinct from beta chain. So we wanted to knock out all the ways of signaling from IL-3, IL-5, and GM-CSF. So we had to make a double knockout of the receptors in these mice that also develop PYM2 ova tumors, and then transfer these tumors now to, again, the system that we have done several times in the past, which is TSLP induction in the context of TSLP-derived deficiency. However, in this case, we are now giving the mice regular wild-type CD4 T cells as the T cells that should be pretty good in responding to TSLP, and comparing them to the TSLP or knockout T cells, which obviously are negative control here. So when we do this analysis, we see that we lose the efficacy of TSLP induction, and there is no difference between wild-type and TSLP or knockout in control of the tumors. We do not see any differentiation in the tumor cells or in the tumor foci. All of them grow out eventually, and there's pretty much no difference between the test and control group. And that really answered this question, this fundamental question, and this is summarized here, that TSLP, by inducing in all of the experiments that we showed, by directly activating CD4 positive T cells and pushing them to a Th2 polarization status, enabling these T cells to release very high levels of IL-3, IL-5, and GM-CSF, which can then directly bind to their receptors, common receptors, if you will, on cancer cells, tumor cells, pre-malignant cells, and promote their differentiation into what looks to be very much a fibrocystic structure with no potential for growth, even after you remove this inductive signal from the breast. So this collection of data really allowed us to arrive at these conclusions that TSLP-activated Th2 cells block breast cancer development by directly inducing the terminal differentiation of breast cancer cells. IL-3, IL-5, and GM-CSF released by Th2 cells are required for breast cancer suppression by direct binding to their common receptor on breast cancer cells. And the activation of TSLP-Th2 axis could represent a novel strategy for cancer immunoprevention. Finally, understanding the role of Th2 cell immunity in early breast carcinogenesis is critical to prevent skin cancer development. This is one of the areas that we are heavily at this point pursuing, with the understanding that there is emerging understanding of the role of Th2s and type 2, if you will, immunity in the breast gland itself, in the normal breast gland development, and trying to understand how that regulation in the normal gland would translate into cancer prevention is really quite fascinating. And again, at the same time, we would love to understand how we could employ some of these discoveries, especially induction of TSLP in patients who are having to deal with very early stage of breast cancer development, and could we deliver this inductive effect and help prevent the recurrence of the tumors in the patients who are getting surgical treatment of the early breast cancers. And we are actively engaging in that process, I should say, in context of trials, looking at induction of this cytokine between the time of core biopsy to diagnose the breast cancer and removal of that lesion that is, again, deemed to be surgically resectable tumor. And with that, I would like to close by thanking our collaborators, Dr. Leif Ellison and Michelle Specht at MGH, member of the laboratory, to participate in these studies I described, Dr. Segroy and Dr. Cohen, again, on the side of pathology and also immunology that collaborated with us, Dr. Liu from Roswell Park. His team has been helping us with the in bioinformatics analysis of the data that I presented, and funding from IOTN that really enabled these studies into breast cancer immunoprevention. And with that, I would like to thank everyone for joining in. I'm happy to answer any questions. Thank you so much.
Thank you so much, Sean. Very nice talk and incredible data and presentation. So I think there is already a couple of questions in the Q&A chat box, but let me ask you first a quick question. Do you have any plans to move this forward in immunoprevention or clinical trials?
Yes, exactly. So the neatest, or what we think is the node for the activation in those pathways, seem to be very much TSLP. In other words, if you induce the cytokine, we get the full picture of protection. We have been trying to look at these other downstream cytokines as well to see if their induction alone in combination would substitute for TSLP. But in experimental model, preclinical model, every time we do this, we come back to best efficacy is really delivered by TSLP. And that could be simply because it's upstream activator of the whole pathway. And for that reason, we actually have tried this concept already in the clinic using very much what we discovered and demonstrated in patients for skin side of things, which was topical, if you will, topical induction of TSLP with the same vitamin D analog. And still work in progress, but there might be challenges with that with regard to systemic availability of TSLP after a topical treatment, unlike in mice. Humans may not, although skin expresses it, but may not get much systemic to allow for the presence in the breast microenvironment. So we are actively, actually with the PREVENT program at NCI, we are really trying to understand what could be the alternative way of delivery, mRNA or other ways that we could deliver this cytokine into the breast and directly ask if we locally express this cytokine in the temporal fashion, again, between the time of the biopsy and excision of that breast tumor, do we get induction of CD4 T cell immunity? And do we see any alteration in the breast phenotype, the cancer phenotype itself? So yeah, that's very much an area of, for us, huge interest, because again, we have seen the efficacy of this in context of skin cancers, and all the experimental data suggests should have a similar efficacy in terms of prevention in the breast. But again, the technology still needs to be developed. It's not as easy as topical treatment for skin cancer.
So it sounds like your guys are still working on optimized delivery methods.
Yes, correct.
And in parallel, I guess that, thinking on implementing this in future clinical trials, is there any activity into identifying how this may play, the entire pathway may play in different patient populations? I am thinking in particular with high-risk populations for breast cancer, in potentially if what will be the effects or in BRCA1 or BRCA2 carriers, things like that, right?
Yeah, so that we are currently testing that in the animal models. So we have generated the BRCA1, BRCA2 mutant models, or obtained them and reestablished them in our laboratory. And that's where that stands in terms of understanding. That's obviously a very important question that we need to get at in context of prevention beyond looking at, if you will, early malignancy. But that's work in progress. We don't have a good handle on what the effect would be and how effective this cytokine signaling would be in that context. Okay.
All right. Well, we can go over some of the questions that we have in the Q&A chat box. First one is from Dr. Hartman. Did TSLP expression also alter CD8 positive or antibody responses, quantity or isotype against OVA in your models?
That's a great question. Definitely, that effect did induce CD8 immunities, again, as a minor contributor, we believe, because of the study that we did, which was, I didn't show that here, but we made mice that overexpressed TSLP in the PYMT spontaneous models and in a RAG1 knockout background. And in this context, if you then, obviously the protection is gone when you do that, when you remove RAG from mice. But then when you put back CD4 T cells, you regain that protection against, for the TSLP against breast cancer. You don't gain that back if you put back CD8 positive T cells into mice. So on their own, in responding to TSLP, does not seem to be very effective, but we believe it's contributing to what we're seeing downstream of CD4 positive T cells. In context of antigen specificity of those CD8 T cells or B cell antibodies, we haven't really pursued that. That's an amazing question, and we do want to understand that, especially for B cell side, because we're looking at T helper cells. I want to understand how that B cell compartment is contributing to this, but that's still a work in progress.
All right, another question that I think can be answered at the same time for two of the attendees. So does TSLP signaling occur at metastatic sites like liver or brain? And similarly, Paul Hennick and ask, have any other tissue cells shown similar responses to TSLP?
Correct. Yeah, great question. So TSLP in the context of advanced disease, it's a little bit more nuanced conversation, which is what would be the effector cell or the cell that is receiving the signal from TSLP? There are very solid data arguing that in the context of breast cancer cell lines, modeling advanced breast cancer, that perhaps those cold tumors, if you will, the tumor cell lines that develop in context, in absence of T cell availability, the TSLP signal that might be present is really recognized by myeloid cells. And that could be pro-tumorigenic instead of being anti-tumorigenic. So what we are, and we agree with that, what we are saying in many of our models is that in this early cancer development, because you're dealing with a not very immunosuppressed, if you will, tumor microenvironment or tissue microenvironment, you have permissiveness of TSLP signal being received by T cells and giving you protection through the T cell axis, CD4 T cell axis. So if I had to go back to the metastatic models, the best data we have is baseline TSLP in a metastatic tumor model might not be protective. It might be actually tumor promoting. However, if you bring induction of TSLP into the picture, so with the systems that we have or locally induced TSLP in those contexts, you can induce recruitment of CD4 positive T cells and gain some protection from that axis. But we believe in all of the studies that we have done, it has been really the TSLP to CD4 axis that was the key axis for protection in this context. To the other tissues, yes, we have seen, as I described, initially we discovered this in the context of skin, and then more recently we have also shown similar pro-differentiation phenotype in lung cancer as well. So again, for these epithelial cells seems to be, at least in early phase of their development, they're responding to these signals very similarly across the board. But those are the areas we have looked at: skin, lung, and breast.
All right. We have another question. Is beyond CD4 T cells in the breast epithelial cells, does TSLP induction also increase dendritic cell or macrophage mobilization trafficking?
Yes, yeah, definitely, yeah. Especially for disease, yes. But when we see these, and that's pretty much follows how TSLP works in context of allergic inflammation. In case of macrophages, we see expansion of them. We don't really know how direct that is to the induction of TSLP versus, again, downstream cytokines like IL-3 and GM-CSF and other things that are coming off of CD4s, and that way they're expanding the macrophage population.
Okay, moving forward. Sounds like there is a question from Susan McCarthy. I am not totally clear on whether you always block IL-3, IL-5, or GM as a group, or look at their individual effects.
That's a great question. So actually, we started by looking at their individual effects, and we couldn't see anything in terms of individually blocking them. And that's obviously, we knew about the fact that they have common receptors and might be, covering for each other, and there's some redundancy in there. That's why we ended up eventually going into the complete 3 cytokine blockade to look at the phenotype. So each one individually did not reverse the effect of TSLP.
It sounds like there is another question here that is, I think you partially or answered when we were discussing high risk population. So from JB, so if I understand correctly, this methodology is curative. Is it applicable for all types of breast cancer? So it sounds like we were discussing this in the context of high risk population, right? But I mean, I think this question can be also interpreted in the context of the different subtypes of breast cancer, right? Triple negative or ER,
Yes.
all of that.
Yeah, so I can say that all of this work so far is in animal models, but we have demonstrated similar phenotype on primary breast tumors from PYMT mice, from HER2 overexpressing tumors, and also from BRCA1 deficient tumors. What Eduardo pointed out previously was the question of taking this technology, going backwards at the point that the BRCA carrier patient does not have tumor, can we intervene and see a similar phenotype or protective effect? That we haven't really done yet, but we have done on primary tumors, and we see a similar reversion to differentiation in the tumor transfer models from these various genetic mutation models of breast cancer. And it globally looks very similar across the board. And again, many of these tumor models in mice are triple negative light. They don't have ER receptors, so we haven't really been able to really look at ER positive or hormone receptor positive tumors. But we think again that when we look at this axis and the receptor expression on human tumors, we see these cytokine receptor expression across the board in the patient's tumors and breast glands. So we believe it could translate quite well across into the ER positive. And that's where, if you were going to go for the early breast cancer therapeutics to prevent recurrence, ER positive probably would be one of the primary places we will be looking at.
We're getting into more questions here. It sounds like you use TSLP-R knockout mice for the tumor study. Can you please provide some information using the wild type rather than the TSLP-R knockout mice?
Perfect question. So we have done this in our previous publication in 2016 when we, for the first time, demonstrated that TSLP induction has a tumor protective effect in the breast. In that context, the mice were wild type. What I'm demonstrating or showing here is really a mechanistic study, which was to understand what just happened. Because as you can step back and look at this discovery, many steps of it is sort of very unconventional as we think of cancer immunology. So you look at Th2 cells and differentiation versus CD8 T cells killing tumor cells. And we really wanted to focus on honing on the mechanism here. That's why all the mice I present here are receptor knockout, just to clean up the system, only allowing one cell type to receive the signal. But prior to this, we have demonstrated that if you just induce TSLP in wild type mice, you get a very robust tumor protective effect.
There is another question from Paula Ganek. Could this be used as a tumor site agnostic chemopreventive therapy? I guess that is linked to, have you tried this in some other model or some other organ?
Yeah, I would say, again, for skin, lung, and breast, yes. But outside of that, I would maybe argue, again, we don't have any solid data, but maybe argue that for epithelial cells, epithelial-driven tumors, we think we could make the argument that epithelial-derived alarmin like TSLP could have a similar efficacy induction of CD4 T cell immunity. But we haven't really tested those. But in the three organs I mentioned, we definitely have solid data. And again, in the skin, we do have immunotherapy based on this discoveries that now it's being used clinically worldwide. So hopefully, again, our next stop is breast, and see can we take these discoveries into the clinic and help patients.
All right, lots of good questions. There is another one here. Why wild-type CD4 T cell transfer only protected a fraction of the recipients? Are there any differences between the CD4 T cells in protected versus non-protected recipients?
That's also a great question. We connect that mostly to the fact that these tumor models are primary tumor transfers, and the heterogeneity might be the factor in why some tumors respond better than some other tumors. But we would like to go back. That's a great question of going back and seeing the ones that are not completely responding and going to complete reversal. Is that tumor cell dependent effect, or is it the transfer T cells that are coming in that are not really homing into the gland or into the tumor? And that leads to that lack of efficacy for those tumors. We haven't really gone back and looked at that because in many of these cases, we followed the mice out for many months afterwards, focusing on the ones that are protected. That comparison hasn't been enabled, but that's something that we will be looking at going forward. Yeah.
Another question from Denon Wang. Certain glycan markers may be triggered to express with terminal differentiation. Is there anyone looking into it?
That would be great if you could look into it. We haven't looked into that, but it's a great question. We certainly can look at that. We have the samples, and we looked at classical markers as I showed here in this presentation, but definitely some of the other markers you associate with differentiation could be looked at here, and the prediction is that they will be increased upon TSLP induction.
Yeah, there is a final question here for you, Sean.
Thank you.
We are grilling you. I mean, this looks like almost a PhD dissertation. But do you find overexpression of other cytokines such as TNF alpha?
Exactly. That's actually a fascinating question, and the short answer to that is yes. So, and this is actually a good, very nice last question, which is how do you describe this Th2? What is really at the core of this discovery? And why this discovery perhaps is very distinct from textbook way of with what we think about Th2 cells in cancer immunology and how we think sometimes of it as being tumor promoting. We are understanding, and it's an understanding that's emerging in the field, that perhaps there are two types of these Th2 cells. One type would be regulatory Th2s that have more of that regulatory function and more of a, if you will, in tumor context, tumor promoting function. But there's this other type, which I like to use the word inflammatory Th2 cells, which what we are seeing in this context. And they're putting out massive amount of cytokine, again, on the conventional Th2 cells, but also IL-3 and things that you think of mostly in bone marrow and like in hematopoiesis, that they're generating loads of them. And one of those cytokines among these is actually TNF alpha. And I didn't get a chance to show that data. We looked at TNF alpha and also interferon gamma for that matter to see if those would have any effect in the phenotype you're seeing. They're not really delivering any efficacy if you remove TNF alpha from these CD4 T cells, use TNF alpha deficient or gamma deficient, interferon gamma deficient CD4 T cells. They're as perfectly able to prevent or revert tumors upon TSLP activation as wild type T cells. However, this conversation that these cells can express the massive amount of TNF alpha might then translate into, when we looked at these things in advanced tumors, how that might be protective in that context. As I mentioned, induction of TSLP in advanced cancer of the breast can still be protective when it engages CD4 T cell response into the tumor. But we don't understand how that works, because in that context, at least in the cell line models, you can't really drive differentiation. We cannot drive differentiation. So we are really trying to understand what is the protective, what is that control phenotype looks like, what is the mechanism, what's the driver? Is it still these cytokines or other cytokines coming from CD4s? That's open question, but just to say that we believe these T cells are inflammatory Th2s.
Probably the last question.
Yeah, so I was going to say we are almost there and wanted to be respectful for everybody's time. So there is a final question from Aurelia Lugia. Does TLSPR mediates actions of other cytokines beyond TLSP?
Oh, yeah, correct. We don't know. We believe that TSLP receptors really only binds to TSLP. So it's a TSLP receptor is a subunit of the actual receptor. The TSLP-R is one subunit, the IL-7 receptor alpha is the other. So the two together bind to TSLP. And so far that is known to be the ligand for this receptor, the sole ligand.
Well, I think that wraps up today's presentation. Sean, thank you so much for an awesome presentation. Very clear, and also thanks to you and all the attendees for the phenomenal questions. I think the Q&A session was as enriching almost as the talk.
Yeah, so thank you for moderating it. It was great. Thank you for taking care of it.
Yes, also thank you, Dr. Villar-Sanchez, for moderating. That was a lively Q&A session, and thank you, Dr. Timihiri, for an excellent presentation. Just like to remind everyone, our next Moonshot seminar is in November. As you can see there, Dr. Gumas will be talking about development of a visualization approach to enhance cancer moonshot data. So thanks, everyone. See you in November.
U.S. Department of Health and Human Services, National Institutes of Health, National Cancer Institute, cancer.gov, 1-800-4-CANCER.